Mohammad Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS for the welcome address. Please, sir.
Good morning and good afternoon. Assalamu alaikum, ladies and gentlemen, boys and girls, delegates and scholars. And this very important event, this is a series which we have initiated about a year ago, in which we are inviting top scientists, very high caliber scientists of different disciplines to deliver lectures. And the objective of this lecture series is to expose younger people with the best of scientific development taking place in different disciplines. Today is something exceptionally important because we have someone who has contributed all along her life in the field of natural product chemistry, pharmacognosy, and pharmacy. And she has made tremendous contribution. She has trained many people in her discipline. She is the teacher of teachers, and you will find many of her graduates who completed their PhDs under the supervision of Professor Dr. Bilge SENER in top positions all around the developing world and also in developed world as well as in Turkey. Professor Dr. Bilge SENER is going to speak about a very important topic which is related to drug discovery and development, and that is the bioactivities of secondary metabolites from Turkish medicinal plant. Professor Bilge is from a top institution of Turkey, which is called Gazi University, and she's also an adjunct professor at the HEJ Research Institute of Chemistry in Karachi, Pakistan. She is a scientist of excellence and commitment. She's very much motivated to spread the knowledge, and she has been always forthcoming to help people from the developing countries. She has visited many countries of the world and has helped many institutions in their development. Professor SENER graduated from Ankara University. As we know, Ankara University is the top institution in Turkey in 1974 and completed her PhD, awarded by TÜBİTAK. And TÜBİTAK, as we know, is a premier research establishment in Turkey, associated with Ankara University in 1977. In 1981, she became an associate professor at same department and later become a full professor in 1988. She has been to different places of the world, including Department of Chemistry, Pennsylvania State University, where I have had the honor of working with her in the same lab. And these were the memorable days where we were able to do lots of good science and of course develop that long friendship, which has resulted in publication of several hundred research papers with the scientists of Pakistan. Professor SENER is also involved in several administrative tasks. She was the dean, she was the co-director of various faculties in Gazi University. She has been the Turkish delegate at European Pharmacopoeia Commission by the Ministry of Health. She has also served as a member of the Bioorganic Committee of IUPAC, which is International Union of Pure and Applied Chemistry. And her main research field throughout her life has been the development of bioactive compound from the medicinal plant and largely from Turkish plants. She has worked on the evaluation of herbal medicine along with nutraceutical for their quality control. She has won many international awards, including national and international award, about 21 of them. And in addition, she has completed 48 projects supported by NATO, National Science Foundation, IUPAC, Socrates, Grundtvig, TÜBİTAK, and University Research Fund. She has authored and co-authored 16 books, over 106 chapters, and 719 research articles, which represent very high level of excellence and the breadth of research she has been able to contribute. She's also given 71 conference lectures and 358 plenary and invited lectures in international conferences. She has supervised 22 PhDs at Gazi University and has been the co-advisor of 16 PhD scholars at University of Karachi. Most importantly, the international character of Dr. Bilge contribution is extremely important because she has been a global citizen, has worked with many institutions and benefited the humanity in major way. It's an honor for me to invite my very dear friend, Professor Dr. Bilge, for her presentation.
Thank you very much, Iqbal. Actually, it's very kind to accept for me your very, very kind invitation. But in the webinar, unfortunately, not face-to-face, but it's also good to see you also, the other colleagues, not face-to-face, but on the computer. And also, thank you very much for your very, very kind introduction. And this afternoon, I would like to talk about our bioactive compounds, particularly isolated from Turkish medicinal plants. And I would like to give you some brief information about the importance of them, healthcare, and some ongoing approach for natural products and bioactive compounds obtained from our resources. As we all know that plants are livestock which supplies individual needs as foods, clothing, and shelter, as well as healthcare, to formulate some pharmaceuticals and cosmetics, perfumes, so on. And they are utilized by people of homeopathy, allopathy, Unani, Ayurvedic, along with some Chinese medicine to treat the diseases around the world. Of course, we need sometimes new drugs, depends on different difficulties on our global healthcare. And as we all know that population is increasing in the world, technology, okay, is increasing, is the best.
But Bilge, would you like to share your presentation? Because I think this presentation is not shared yet.
Really?
Yeah.
Oh, sorry. I have already shared. I don't understand what happened.
I think you logged out after that, so it canceled. You can try sharing it again.
I have to cancel first?
No, just share it like you did before.
Okay. So did you manage?
Not yet, it's not.
Steve, can we do it from here?
It's starting to share now. Yes, it's
visible.
visible.
Okay.
Excellent.
It's visible.
Excellent.
Okay. Oh, I think I have to start maybe first. All right. Sorry. And I would like to give you some brief information about healthcare products and some approaches on natural products and some examples from our studies on medicinal plants growing in Turkey. As we all know that plants are a livestock which supplies individual needs and as foods, some clothing, shelter, healthcare, as well as pharmaceuticals throughout the planet. And they are utilized by people of homeopathy, allopathy, Unani, Ayurvedic medicine, along with Chinese medicine to treat the different diseases in the world. And increasing of the population and increasing of the technology, but we have also face-to-face increasing some killer diseases. As we all know that nearly 2 years almost, we have a problem, this COVID-19 diseases. There is no specific good medicines. Of course, we have some vaccines, but vaccines is not sufficient to stop these diseases. So we need some new antiviral compounds. And also we have to need some new drug candidates for other diseases. And we have to face oil supplies is decreasing, and we also lost our biodiversity to destroy all of bioresources. So development of traditional medicines is very important to safe and effective agents, and discovery of new drugs for existing new lead candidates. So natural products also present challenges for drug discovery, such as technical barriers to screening, isolation, characterization, and optimization contributed to a decline in their pursuit, the pharmaceutical industry from the 1990s years. In recent years, of course, there are several technologies and scientific developments, including improved analytical tools, genome mining, engineering strategies, along with microbial culturing advances are addressing such challenges and opening up new opportunities. And from the opening of 20th century, allopathic systems of medicine have popularity among the people, which is based on fast therapeutic actions of synthetic drugs. However, traditional route of healthcare system is recently shifted from synthetic to herbal medicine. To universal trend is called to return the nature, or in other words, we call it rational phytotherapy. But we have also problem to use for traditional medicines for their safety and efficacy lacking, as well as some adulteration, different compounds. So in the world, there are several institutions engaging natural product chemistry started to find cheaper and improved remedies for existing diseases like COVID-19. And also due to some side effects of current medicine, we need some new drug candidates. And some diseases have no good remedies during this time. For example, some immune diseases also we have no good medicines. And we have also face-to-face to find some new antibiotics, depends on the occurring resistance of the microorganisms. Therefore, natural products or natural product chemistry is very, very important in this field in terms of drug discovery procedures. Of course, there are different approaches for discovery of plant-derived drug candidates. And firstly, we have to find, identify the biological target. Biological target usually comes from ethnopharmacological data. Of course, phytochemical diversity, chemotaxonomy, and also some the other secondary metabolites used different structural diversity possible to identify the biological target. But the best way to use ethnopharmacological data, because the traditional medicine is also important, used since several years from the human, looks like a pharmacovigilance studies is available. Of course, computerized literature surveillance is also another way to find the biological target. And in vitro biological evaluation of plant extracts mainly used cell and mechanism bioassay for them. High throughput screening is a major strategy to discover new lead candidates from nature's bioresources, not only plants, by using humans, animals, or some microorganisms, as well as marine organisms. So biochemical targets of interest in pharmaceutical lead discovery range from enzymes and receptors. And this ethnomedicine is very important, I said, because in the planet there are approximately 275,000 higher plants is already identified. And among them, nearly 15,000 of them have ethnomedical reports. 15 of them ethnomedical defense. That means the 15,000 is belong to medicinal plants. So we can choose as a biological target for this ethnomedical, according to the ethnomedical reports. And the other hand, approximately 60% of the plants, is 15,000 of plants, not reported chemically or biologically. And another reality, 50% of the plant-derived drugs is composed prescribed drugs used by human literature. If we can give some examples, for example, morphine obtained from poppy species, and some digitoxin, it's the one, and there is no any alternative medicine to compare digoxin for the treatment of cardiovascular diseases, and cocaine. Natural products and their structural analogs have historically made a major contribution to pharmacotherapy, especially for cancer, like vinblastine, taxol derivatives, as well as some infectious diseases, some artemisinin to use for the treatment of malaria. And of course, some other new generations of antibiotics is also available for the treatment of infectious diseases. Therefore, drug discovery and development of new drug candidates for necessary for human healthcare have during this world. And these secondary metabolites not only as a used drug candidates, but also starting material to produce some semi-synthetic compounds used in the treatment of cancer, like etoposide, teniposide, or irinotecan, topotecan. And these natural compounds or bioactive compounds is also template for synthetic compounds. And now we have several antiviral drugs used in human healthcare, but as we know that none of them is possible to use for the treatment of these COVID-19 diseases. Of course, there are different mechanisms involved, like S-protein, M-pro modulators, along with AC2 inhibitors as potential therapeutics. But I think I have heard during the last week, FDA has already approved one of the antiviral product called molnupiravir, is already approved by FDA and also in Great Britain. maybe in next year, we have several antiviral compounds for these pandemic diseases. And during our studies, as approximately, because Professor Iqbal already mentioned, we have started to work on them in the beginning of 1980. And until now, we have is almost nearly more than 30 years on medicinal plants. And during our studies, we have obtained some bioactive compounds from different secondary metabolites. Some of them belong to nitrogen-containing compounds, comes from amaryllidaceae, isoquinoline boxes, and steroidal type alkaloids. Also phenolic type compounds, flavonoids, lignans, coumarins, and some terpene type compounds. As we all know that drug discovery and development is not easy because it needs some several steps, and starting discovery and then doing some chemical and biological characterization. And after that, this is the most important part, is belong to preclinical studies and formulation. And of course, clinical studies. And after complete the clinical studies, it is not difficult to arrange regulatory procedures and marketing. But mainly the preclinical studies in terms of for safety and toxicity using animals is the starting point. Of course, we have to formulate of these drug candidates because we cannot use as a pure state. And we have to arrange the doses. Doses is also very important for individual persons, and dosage forms, as well as some of the bioequivalence, bioavailability studies. And during this time, also nanomedicines are used for healthcare systems. Clinical test takes a long time because this several steps, phase 1, starting phase 1 until phase 2, phase 3. And phase 4 is after post-registration monitoring time. But phase 3 takes really a long time. It takes several volunteers, more than 20,000 volunteers and different countries. So regulatory procedures is already done. Marketing is important because during the marketing, some of the drugs have given different actions, positive actions, as well as some adverse effects. And this marketing surveillance, we call it pharmacovigilance, is also important. And during these discovery procedures, this one of the aspects is enzyme inhibiting activities, because we all know that enzymes play an important role in all aspects of healthcare. For their overexpression and hypoactivation is involved the pathogenesis of different diseases. And another step, we have to use this kind of enzymes is originated from human enzymes. And during this test, in vitro biotest, we have to use some reference compounds for the beginning stage. And if we look at the mechanism of different diseases, is also involved like anti-inflammatory and anti-diabetic and anti-cancer, as well as other diseases have some strategies, depends on the enzyme inhibitory theories. So enzymes interact with different inhibitors by blocking their activity towards corresponding physiological correspondings for natural substances. So nearly 20 years, we changed our studies towards the neurodegenerative diseases, because neurodegenerative diseases is also very, very important. In our country, we have, of course, very rich flora. Is about 13,000 of plant species growing in Turkey, and 800 of them will already reported medicinal aspects. And during these works, we use activity-guided fractionation, not bioactivity, activity, because we use the in vitro screening test. And we have done some synthetic works and structure modification, and some of them is also used in preclinical studies, but not completed because we need some multidisciplinary works on that. And as we know that neurodegeneration is characterized by functional loss of nerves, finally leading to neuronal death, and it involves several diseases starting demands type of health problems. And if we look at the incidence of these diseases, and the number one is Alzheimer's diseases all the world. And Alzheimer's disease, there is no specific mechanism is responsible for the treatment of AD, but several of mechanisms. One of them is anticholinergic drugs, and we use just only maybe 3 or 4 drugs is available during this time. And also several mechanisms are involved Alzheimer's diseases, like amyloid proteins and other as. And enzymes also play an important role for these aspects. And all the drugs is available on the market is belong to anticholinesterase type of compounds for the treatment of Alzheimer's diseases. These are 2 sister enzymes, actually, cholinesterase inhibitions. And one of the products is memantine. It belongs to different mechanisms. Is like amyloid proteins, inhibition of the collection of amyloid proteins. And some drugs is helpful, but not completely treatment. And there are many clinical studies on ginkgo biloba for cerebrovascular circulation increasing drugs. But so we concentrated these anticholinesterase type compounds to search the medicinal plants growing in Turkey. And acetylcholinesterase and the other sister enzymes is butyrylcholinesterase. And there is also a very nice modified Inman method used for in vitro screening test. And the results is also informative, corresponding used as reference compound. We actually used a reference compound. This is also one of the drug, grantamine. Look at here, there are very, very few, and there are no more drugs is available on the market, like antibiotics or analgesics or anti-inflammatory type of drugs. But, so we need some new and good medicines. So in the world, if you look at the literature, there are many, many works on this. Every day I got several papers from these mechanisms and new lead compounds from natural sources. On the other hand, if we go to the ethnopharmacological data, we have seen several medicinal plants used in ancient times for the treatment, not treatment actually, to enhance the memory, enhancement of memory to helpful Alzheimer or other demands types. And one of them is a Chinese plant, Persea, and is already formulated piperazone. And there are several clinical studies done, not only in China, also in the United States, but none of them is completed. In Europe, there are several species like salvia, rosemary, corydalis, laxus, used memory enhancement procedures. So therefore, in our country, as I mentioned, more than 13 plant species growing in Turkey, but 800 of them is already reported at medicinal aspects. And we have is about 2,400 of them is already screened for these anticholinergic activities. And in it's about 1,145 taxon among the 13,000 plant species is belong to bulbous plants, and these plants, some of them is cultivated as an ornamental species to export as an ornamental purposes, but we checked them for anticholinergic activities. Of course, these species, some of them is very few taxons, but some of them is very, very high amount compounds. So this project is actually completed 5 years, and we have collected all of them from the country, and also they preserved in the horticulture center of Atatürk Arboretum near Istanbul. And this is the in situ cultivation. Because we have to also keep our flora, because Turkey is one of the belong Convention of Biodiversity Pact. So we have to collect not huge amount, but small amount, and then we have to cultivate them. So these bulbous plants is very nice ornamental flowers, but they have also very nice bulbs underground. And these bulbs, some of them is very small, some of them is a big amount, but we have collected all of them and then screened by using in vitro anticholinergic activity. And from Amaryllidaceae plant, Galanthus species, we have started, of course, Galanthus species, it depends on the galantamine drugs, and Narcissus, Leucoium, Pancrasium. And at the pre-screening test, they gave us very, very promising results. So this among them, Galanthus species, as well as Narcissus tazetta, gave us some pure active Amaryllidaceae type alkaloids. From them, lycorin is very, very high inhibition. IC50 is about 3.16, and this is very nice compound, Amaryllidaceae type. And but we need to synthesize and to prepare some derivatives for structure activity relationship studies. Not all of them is completed, but we have also started them. And if we look at the IC50 of galantamine, 3.2, so it's almost a similar type of activities. And then we searched Lycopodium species, depends on the Hypersia, is the Chinese plant. But in Turkey, we have no Hypersia species, Hypersia serratum actually, but the other synonym of species, Lycopodium, because Lycopodium serratum is the synonym of Hypersia serrata, is one of the traditional Chinese medicine used memory enhancement properties. Unfortunately, we didn't put hyperzine in our Lycopodium species. These are the pteridophyta plants. However, we have find one of the compounds, alpha-onoserine, is very promising activity if we compare the lycorine as well as galantamine. And alpha-onoserine, after structure elucidation, this compound is not belong to nitrogen type containing compounds and different structure. So it's also one of the candidates for further studies. And we have to check huperzine A, IC50 is 1.6, galantamine is 1.8. So huperzine is really, really nice. And I'm expecting this product for the treatment of Alzheimer's diseases. And also we have prepared some other derivatives from galantamine and some derivatives like N-alinor galantamine and also N14-methyl allyl norgalantamine is very, very high IC50 values. So therefore we have to find drug candidates, but we need also some to prepare derivatives to compare the activity. Maybe some derivatives is more stronger activity than. And depends on the ethnopharmacological data, particularly in European countries. Salvia species Rosmarinus stellaria is used for traditionally memory enhancement purposes. So we decided to check also aromatic medicinal plants for their anticholinergic activities. Salvia sage reported also use memory enhancement. In Turkey, we have 96 species growing in Turkey. We collected all of them and identified all the species. And we have checked their anticholinergic activities as well as antioxidant activities. Of course, they show a very high percentage of antioxidant activity. So we noticed that besides anticholinergic activity, antioxidant activity is also supported anticholinergic activity. And you see, I will not give more information because we have already published this data in the literature. And some species, you see the green color, I indicated green color, is very, very high percentage if we compare the reference compound galantamine. And all 19 species, we have also worked, one of them is Salvia triloba, to use in vitro screening isolation. And we obtain some terpene type compounds I will mention later. And rosemary, another plant, is also used in spice in our kitchen. But this species used in Europe in ancient times for the improving memory. And of course, this aromatic medicinal plants contain more essential oil. And as well as we have find one of the compounds, rosmarinic acid, is very high activity. And this, of course, as you know, familiar, this in vitro screening isolation test, we have first started fractions and then isolation, the pure compounds, as well as the other secondary metabolites like essential oil. Essential oil is also essential oil fraction. They have a very nice inhibition corresponding galantamine. And other compound, rosmarinic acid, is one of the phenolic type compound. It's also very, very high percentage anticholinergic inhibition, but not acetylcholinesterase, another sister enzyme, butyryl cholinesterase. And as you see, it's very, very simple compound, rosemary acid, and it's available on the market. And maybe to prepare some derivatives, make some stronger inhibition activities and some structure activity relationship studies is also important. And after these phenolic type compounds, we thought that some other, as well as antioxidant compounds like flavonoid derivatives will be possible anticholinergic activities among secondary metabolites. So in this purpose, we have worked on Maclura pomifera and other medicinal plants. It's a very nice and very big fruits, and it contains flavonoids, several types of flavonoid compounds, and two of them actually, quercetin and macroxanthin. And particularly macroxanthin gives very, very high inhibition comparing galantamine. And we also extend our studies to support in vitro data by molecular docking studies. And this macroxanthin gave us the best confirmation, exhibited the tight binding, particularly in vitro and docking studies, and this data is supported our in vitro binding assays. And during this kind of flavonoid type compounds, we have also concentrated one Angelica species, and this plant was containing some coumarin type of phenolic compounds. And during our activity-guided fractionation, we obtained imperatorin, xanthotoxin, and bergaptan. This, of course, known compounds. It's not new one, but it's a very nice inhibition comparing galantamine as a reference compound. And these three coumarin derivatives is also studied to obtain, to calculate the docking energies, and it can be concluded that these compounds are potent inhibitors of the target of human butyrylcholinesterase activity. And as we know that Rosa damascena, known rosemary, used in medicine since ancient times. And also Avicenna, if we look at the old literature since Dioscorides and Ibn Sina, we noticed that this rosemary against different diseases, but as well as during Ottoman Empire, is described make brain stronger, because that time they have no idea about demands, but to make brain stronger or enhancement of memory. And in Turkey, we have cultivated Rosa damascena, and we produced rose oil to export. And we checked this rose oil, essential oil, and also the aromatic water of Rosa damascena, and supporting by GCMS studies, and we have obtained several fractions from this essential oil and aromatic rose waters. And also we have isolated some major compounds from the essential oil like geraniol, nerol, stronellol, and one of the compound, this is aromatic compound, phenylethyl alcohol, gave us very, very high percentage of inhibitory activity. So this result supported the traditional uses or ethnopharmacological data. And we have prepared some different proportions of these promising terpene type compounds with phenylethyl alcohol. And one of the mixture of these compounds give us very high percentage of butyrylcholinesterase inhibition. Is about 84%. And during docking studies to support the inhibitory activity, and we have seen that the docking and binding energies showing that these compounds binding specifically towards the butyrylcholinesterase and acetylcholinesterase human enzymes. And it's after that, because we checked these promising results, we have to extend our works to essential oils, because in Turkey we have several aromatic medicinal plants, and another project we have done on these plants with Konya Selçuk University and Department of Biology, and we have collected all aromatic plants, and some of them, like Melissa, Mentha, Ocimum, Origanum, like Salvia species, and their terpene type compounds, they have obtained activities. Unfortunately, some of them is very, very less activities, but I just only give you some strong activity like Melissa. If you look at this column, you will see the very high amount of inhibition corresponding to the reference compound galantamine. So there are plenty of work on them, and we have also extend our works on pure compounds. These are the terpene type compounds obtained from the essential oil, and some of them is looks like responsible from this activity. But we also noticed that some synergism is also important. The essential oil may be more high inhibition than pure compounds. Another promising aromatic compound, Carvacrol, is about 70% of inhibition. And this essential oil containing medicinal plants, another way to extend our studies on the cholinesterase inhibition. Of course, you need some time and also people to extend this kind of works. And after we have seen that galantamine is nitrogen type compound, so we also extend our works for nitrogen containing plant species. And one of them is Fumaria species. In Turkey, we have 14 Fumaria species, but some of them is endemic species, very rare amount, and a very small amount of this plant. If you look at the plant is very, very small amount. But one of the species from Fumaria variante is also a high percentage collection, and we have done our in vitro screening bioassay isolation tests on Fumaria variante. All of the species, the Fumaria species, if you look at the very, very high inhibition. So this result is also compare our think about the nitrogen type compounds is very, very nice. Is the main drug of galantamine. And after the isolation by using activity-guided isolation, we found, of course, several isoquinoline type alkaloids from this species, is about 102. But particularly protoberberine type of isoquinoline derivatives show very, very high inhibition in terms of anticholinesterase activity. And we also decided during our bulbous plants, Fritillaria. Fritillaria is a very nice ornamental plant, but the bulbs containing nitrogen containing compounds, steroidal alkaloids, and we obtained imperialin as a major compound, and we have also prepared some derivatives comparing more or strong activity than imperialin. This is one of the imperialis, Fritillaria imperialis. But in Turkey, we have about 41 species, and it's not possible to work on them. It depends on some of them is very, very small amount of bulbs, but some of them is reasonable size. But one species, Fritillaria mykolowski, grown in eastern part of Anatolia, and we have done this. We have to start this work with Professor Iqbal Chowdhury group and Dr. Jan Wang particularly worked on this species, and we have obtained several steroidal type compounds, and because alkaloidal extract is shown as very, very strong activity the other species comparing galantamine, and during among of the steroidal type alkaloids, we have also obtained some of the new steroidal types. If we compare the Fritillaria results, but we have not find good results on another ornamental plant, iris species. It's about 82 species, and we screened all of them, but we have not get good results on this. They shown very high antioxidant activity, but there is no results obtained from Fritillaria or other Fumaria species. And these nitrogen-containing compounds is supported our data to extend our studies on other steroidal type compounds, because since 1990, actually, we have engaged phytochemical works. There is no that time biological screening. Phytochemical studies with Professor Ataur Rahman and Professor Iqbal Chowdhury group, and we have actually more than 46 new steroidal type compounds, and there are very, very less amount. Just only we are able to screen and to get a good results. Two of them, buxabenzamethionine and buxamethine, these are also needs to synthesize because their results are also supported by docking studies, but we need some in vivo and as well as some formulation and to extend clinical studies. Actually, it is not possible to work all of them, but we have also worked on other nitrogen containing, particularly isoquinoline alkaloids containing plant species, Corydalis. Corydalis in the world is about 470 species, but in Turkey, we have 17 species with 22 taxons. And we have collected all of them, and we have also kept as in situ in the Atatürk Horticultural Center Research Institute in Yalova. But unfortunately, this plant very, very small amount of bulbs. Using this kind of activity-directed isolation, and we have found, and we did also SCQ12MS studies on this plant species, depends on the amount. And we have collected all of them and with different locations, and we have 51 samples from 17 species. And if we look at the results, some of them is very, very high percentage of activities. And we have also extended our studies by using activity-directed fractionation. Depends on the small amount. We have checked all fractions by using execute of MS. And at the end of these studies, if you look at the material is very, very small amount. So during this extensive execute of MS studies, we are able to screen test fractions. And these fractions also we check the activities, as well as we check also these fractions from the point of chemistry of them. And some of the fractions have very, very good IC50 value in terms of acetylcholinesterase and butyrylcholinesterase activities. And also we obtained at the end of these studies, protoberberine type active alkaloids have very, very strong inhibitory activity on them. And after the isolation of alkaloids from this species, we have found protoberberine type fractions has very, very strong activities. And during execute of MS analysis, and this is the composition of these alkaloids, and some of them is a major compound is seen here, and some of them is a very, very small amount. And this technique is really nice, and we have also identified these compounds, and we are able to check them. I didn't mention the detail because we have already published this data in the literature. So we found the major compound is belong to dehydrocorridolene. And this also shown very, very high activity. This is the results of the alkaloid groups and fractions, retention times, molecular formula, and molecular weight according to the execute of results. And some of them is not known compound, but new compound, but depends on their activity is not promising. So we didn't spend time to identify their structures. And these pure compounds, some of them is seen here, the color numbers, very, very high percentage. And after the structure determination, again, protoberberine type compounds have, some of them is both activities, both strong activities, but some of them is just butyrylcholinesterase inhibition. And these are, of course, known compounds, but it's also important to extend to work on, to prepare some maybe more active derivatives and to increase their bioavailability on them. It is also possible. So we have to work on them. And as you've seen, the protoberberine type compounds is also very, very promising, like amaryllidase type galantamine alkaloid. But okay, we have several drug leads, but how can we extend to drugs? Because drug discovery procedures is not starting in vitro and then in vivo, as well as preclinical and clinical studies. So we have to check their safety and their toxicity by using animal data. So it is not possible to work on this kind of species is a just small amount, 3-4 milligram type compound. So we need to synthesize this compound. And then to prepare some derivatives for structure activity relationship studies and for their safety and toxicity data by using experimental animals. After that, if we can get some safe and non-toxic and also active compound, so we need to prepare formulations like pharmaceutical dosage forms before going to clinical studies. So we have to check them bioequivalence, bioavailability data. After that, so this promising drug candidate will go to the clinical test, phase 1, phase 2, and phase 3 studies. So it's a long way drug discovery procedures. It takes time. Early stage is about 8 years, but now maybe 4 and 5 years we can get good drugs like antiviral drugs. So I also acknowledge this TÜBİTAK project. This is the very, very big project, not only our university, but in Turkey also. We obtained from Turkish Scientific Research Council, and I'm very grateful this institution to support our data. And I also acknowledge my two colleagues, now one of them full professor and the other one associate professor in Gazi University. She is also dean of our faculty of pharmacy, and they are very, very hard worker. And the other, my last students also worked on this project. It's a really long time work. It started 1974 until 2020. And thank you very much for your kind attention. I think I have to keep my time. And if you have any comments, I'm very glad to know your comments and your actions. Thank you.
Thank you so much, Professor Bilge. So what we'll do now is we'll open it for questions. So anyone who has questions, just write them down in the chat and I'll read them out for Professor to answer.
So you have to check the chat box.
I'll have a look, and
Okay.
whatever questions come up, I'll read them out to you.
Okay, thank you very much.
Anyone with questions, please feel free. Okay, there's one question.
I
From
think the solubility.
yes. What is your experience in solubility of compounds in drug delivery?
Yeah, the solubility is actually soluble in dichloromethane and methanol. So, but we have to prepare some salts to increase their solubility in aqueous systems, or we have to prepare some to using nanoparticles to obtain nanomedicine. So solubility, I think, is not difficult to improve their clinical studies, because there are many techniques. For example, during this time, nanoparticles is more efficient to prepare nanomedicines to increase their bioavailability.
We'll just wait a couple of more minutes for any more questions. What I'll do after is I'll leave the questions that whatever questions they have, they can email it to me. I'll compile them and I'll send them to you.
Okay.
I'll also share your presentation with them, with all the participants. So
sure.
You can email me your presentation and then I'll forward it to all the participants present here.
Okay.
I think there are no more questions.
Maybe some collaboration with them.
Yeah.
But usually I explain to collaboration with me and Professor Iqbal Chowdhury's group by using very, very extensive opportunities at the ICCBS.
There is one more question here. How can we decide the bioavailability to bioaccessibility ratio in body for newly found drugs?
I think we did some by using experimental animals first time and before going to clinical studies, because bioavailability is very important.
There's another question. Standardization is major problem in MAPS. How to manage this?
Standardization is not difficulties during this time. Standardization is very, very easy. We can standardize all herbal medicines and to prepare some phytotherapeutic drugs. And there are different examples. For example, in our country, is about 35 herbal medicines are having standardized extracts approved by the Ministry of Healthcare. For example, one of them, maybe all but everybody noticed that ginkgo biloba extracts, and there are, of course, there are different trade names, and is useful for neurological diseases like vertigo and other neurological diseases. Is almost one drug, I say that. So standardization is not difficulty, and it's very easy to prepare standardized extracts. It's also important to evaluate the traditional medicine by rational phytotherapeutics.
I think those are all the questions.
Okay.
So in the end, I would like to thank you once again, Professor Dr. Bilge, for sparing out your time and giving this amazing lecture. And I would also like to thank, we have had over 100 participants in today's webinar. So well done to all of them that attended. And thank you so much once again.
Okay. Thank you very much for you and also everybody's.